Hi everyone, Dr. Mike here. In this video, we're taking a look at the exocrine function of the pancreas. Basically, how does the pancreas work to aid digestion of those macronutrients, proteins, fats, and carbohydrates? Let's take a look. All right, to begin, let's orientate ourselves. What have I drawn up? So this big purple thing here, this is the pancreas. In addition to the pancreas, we can see a couple of other GIT structures. We've got the stomach up here, and we've got the duodenum here. So that's important. Here's our duodenum, the C-shaped flexure. It's hard to say C-shaped flexure. C-shaped flexure of the duodenum. We can see our liver is here. Our gallbladder is here, and then all of these various ducts. Now, when we talk about exocrine function, it's simply about an organ releasing chemicals into these hollow ducts. That's an exocrine function. So let's first label these ducts, shall we? Now, it's not quack ducts, it's DUCTS. So this is what we call the cystic duct. This is what we call the common hepatic duct. This is what we call the bile duct. This is what we call the pancreatic duct. And this is what we call the hepato. So hepato means liver, right? Pancreatic, can never spell and talk at the same time. Pancreatic ampulla. And then this thing here, this little opening here, this is called the pancreatic papillae or papilla. All right. In addition to that, you can see this thing here. So importantly, the functional unit of the pancreas's exocrine function are these things in red. They're called acinar cells, or you could actually take a whole group of them, right? And they are our acinar cells. And let's label that here, acinar cells. And they're going to be very important, but you can also see that there's these black cells here along the ducts. They're called ductal cells. So here we've got ductal cells. Now, what I'm going to do, because these aren't just in this one particular location, they're all throughout, or most of, the pancreas. So I'm just going to draw a couple up here in a more simplistic way. And then, so there they are. And then here we're going to have and I'll just draw them up like this, the acinar cells, just to make it easy. And so let's now label these. These are our ductal cells. So there's two major cell types that we need to talk about when we talk about the exocrine function of the pancreas. It's the acinar cells and the ductal cells. Now again, exocrine, the pancreas has both an endocrine function, meaning it releases hormones into the bloodstream, and exocrine function, meaning it releases chemicals into these hollow ducts. Ultimately, these chemicals will enter into the duodenum, and they are very important proteins that play a role in the digestion of proteins, fats, and carbohydrates. That's the major exocrine function. So to begin, let's talk about these acinar cells. What is inside of these acinar cells? So if we draw it up here, I'll get rid of this so I've got more room. You know that that's the tail of the pancreas. So if I look at an acinar cell, and have a look at what's inside, we've got granules, right? Granules. So you've got these little tiny granules, and they're sitting inside of these little packages, right? Now these granules are important because they contain very important digestive enzymes. So they contain what we call zymogens, and they are inactive enzymes, but also active enzymes. So let's first look at the zymogens. So the inactive, the zymogens. So an example of some zymogens is trypsinogen. It's chymotrypsinogen, right? It's carboxypeptidase, isn't that a wonderful name? Peptidase. proelastase. These are the major zymogens, meaning they're inactive. So if something ends in OGEN, you know it's inactive, right? So some end in OGEN, some begin with pro. So all of these, so inactive, 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 inactive. And all of these here are proteases. They're all inactive proteases. That means they break down proteins. Why would these inactive proteases, so zymogens, sit inside these granules in an inactive form? Well, everything's made of proteins. So if they were activated, they would digest the pancreas because they're in all of these acinar cells. So they would digest the pancreas and you'd get pancreatitis. You don't want that. So they sit in an inactive form. We've got other chemicals, enzymes that sit in an active form in these granules. I'll write it like this: active enzymes. And examples of these active enzymes include lipase, colipase, alpha amylase, DNase, RNase. Now there's obviously others, but these are the major ones. So they're in an active form, and you might think, why is it okay for them to be in an active form? And the answer is because, well, you're not going to be breaking, there's no fats in there to break down. There's no carbohydrates in there to break down that's going to damage the pancreas. Things are made up of proteins. Proteins, you don't want to digest those. You want to wait for it to get inside the duodenum for them to be activated. So we've got granules inside the acinar cells, and that's what's inside them. Brilliant. What's inside of these ductal cells, you may ask? Well, let's have a look. The ductal cells, they have predominantly, the main thing we're talking about here is bicarbonate, HCO3 negative. That's bicarbonate. I might write it here just so you know. It's bicarbonate ion, right? But there's another thing in there. There's chloride ions as well. Now, there's obviously water and other things, but they're the two most important things. So the ductal cells have bicarbonate, and the acinar cells have all the enzymes to break down proteins, fats, and carbohydrates. All right, now we can start talking about how does the pancreas do its job. So the stomach is going to be filled up with its chyme. Remember that when you start digesting proteins, fats, and carbs, it forms this liquid material called chyme, and the stomach will squirt it in to the small intestines. Literally will squirt it in. It's going to contain proteins. I'll just write it as P. It's going to contain triglycerides, so that's our fats, right? It's going to contain our carbohydrates, right? So I'll write it as C. So we've got our proteins, our triglycerides, and our carbohydrates. There's obviously other things in there. So by the time you've hit the stomach, you're going to have peptides as well and some amino acids. You're also going to have, so remember this, right? Where should I write it? I'll write it here. Remember that your triglycerides, right? They have this glycerol backbone with 1 fatty acid, 2 fatty acid, and 3 fatty acid, right? And that's the glycerol backbone. So what we want to do with lipases is chop the fatty acids off. Now we only chop 2 of them off, right? So we end up having 3 fatty acids and a monoglyceride. So that's what we're breaking up. So in addition to having the full macromolecule proteins, triglycerides, and carbs, we have some peptides and some amino acids. We have some free fatty acids and a monoglycerides, and we also have some glucose floating around as well. What else is the stomach squirting out? It's squirting out hydrogen ions because that's what makes it acidic, right? So they're all moving through because you've had your meal. They move through the duodenum and then they get into the duodenum here. What do we find in the duodenum? Okay, a couple of important things is we have a group of important cells called enteroendocrine cells. They're called enteroendocrine cells. So enteroendocrine, what does that mean? Well, entero is referring to gut. Endocrine means it releases hormones into the bloodstream. And you've got two types, right? You've got an S cell and an I cell. Now, as these products move through, they start to get stimulated. So what we end up getting is that when you have things like broken down proteins, so peptides and amino acids, and even triglycerides and free fatty acids, right? Not glucose, but when you have basically the breakdown products of proteins and fats, when they come across the enteroendocrine I cells, it stimulates them to release an important, very important chemical, which I'll do in blue, into the bloodstream called CCK. Now, CCK stands for cholecystokinin. Cysto means gallbladder. That's this guy here, right? Kinin means to move, to contract. CCK is a gallbladder contractor, but that's not all it does. I'll talk about it in a sec. CCK also is a potent stimulator of acinar cells. There are CCK receptors on the acinar cells, and it triggers the acinar cells to release all of their zymogens and active enzymes by telling the granules to be released through exocytosis. So it releases them, it releases all the products of the granules into these ducts. And obviously they're going to travel through into the pancreatic duct. They're going to keep moving across. They're going to get to the hepatopancreatic ampulla, and they're going to be released into the duodenum. That's the role of that CCK, stimulating the release of the granules and their products from the acinar cells. And now we've got them all here. We've got all of these things here in the duodenum. That's fantastic, right? Now, importantly, enzymes will function at a certain pH. And if we've just squirted hydrogen ions into this area here, right, that's going to be too acidic for these enzymes to work properly. So what happens is hydrogen ions will stimulate the S cells. And the S cells release something called secretin. Secretin. So I'm going to do this. There we go. It releases secretin. And what secretin does is it stimulates the ductal cells. Secretin stimulates the ductal cells. What do the ductal cells release? Bicarbonate. So we've also, in addition to having all of these zymogens and active enzymes being released, we're now also releasing bicarbonate. And that's good because what bicarbonate does, as you know, is that when bicarbonate comes across hydrogen ions, they bind together to form H2CO3, which is called carbonic acid, which splits itself apart to form carbon dioxide and water, a gas and water. So it neutralizes it for us. Brilliant. So now these enzymes can function at a neutral pH in the intestines. Thank you so much, ductal cells, and thank you so much, secretin, for doing this. So now we need to talk about how these will work. Well, importantly, what we need to do is we need to have a think about the first one here. I wrote it up intentionally on purpose. So when you've got trypsinogen being released, I'm going to wipe this off here. So let's say the zymogen trypsinogen is here. It's inactive, right? It's not doing anything. It can't chop anything. I can't chop proteins up. It needs to become activated. So what activates it is there's a group of cells present, again, in the walls of the duodenum called enterokinase. And the cells release something called enterokinase. And specifically one called enteropeptidase, which activates trypsinogen, right? And it activates trypsinogen into its active form trypsin. So the enterokinase activates trypsinogen into trypsin. And what does trypsin do? Trypsin takes the zymogens, right? So that's going to be chymotrypsinogen, procarboxypeptidase, proelastase, for example, and trypsin will activate them. It says time to become activated. And then those zymogens will become their active form, their superhero form, which includes chymotrypsin, carboxypeptidase, and elastase. And their job is to chop up peptides and proteins into smaller amino acids. Brilliant. All right. What else? Well, we've got alpha amylase. That's easy. That helps chop up the carbohydrates into glucose. DNase and RNase breaks up those nucleotides. That's important. Lipase and colipase. So remember I said CCK not only stimulated the acinar cells, but CCK also stimulated the gallbladder. And the gallbladder contains bile, right? And so let's draw this bile up in purple because I don't have my green marker with me. It tells it to contract. It squeezes that bile all the way through the bile duct. It joins with the pancreatic. And now we've just released bile into the duodenum. And so what we end up doing is when we've got our fats, right? Remember, fats come together in liquid environments. There's a lot of water in these pancreatic juices that we've released. So the fats come together into a big globule. So what we need is to break that up. And that's what bile does. So bile is made up of bile salts, right? And bile salts have like a head and a tail. And effectively, what they do is they help break up the fats into smaller, more manageable pieces, right? Because effectively what you're going to have is you're going to have these bile salts break it up. That's called emulsification. I'm not going to do it for all of them. I have done a video on the details of this, so I'm not going to go through the details, but here are our bile salts. It's helped break down big fat globules into smaller, more manageable pieces so that our lipase and colipase can come along. So what colipase does is it manages lipase. It says, hey, I'm your supporter here. I'm bringing you along. Think of lipase as being the scissors and colipase being the person that works the scissors, right? And so it says, great, let's start chopping these fats up, these triglycerides up into free fatty acids, right? And monoglycerides. Brilliant. So look at that. This is that function. Now, CCK released by the I cells and secretin released by the S cells are not the only way for us to be able to secrete the granules from the acinar cells and the bicarbonate from the ductal cells. We can stimulate their release another way. So remember, during the digestive process, you've got three distinct phases. The cephalic phase, where seeing, smelling, tasting food triggers the vagus nerve to stimulate all these gastric secretions. You've got the gastric phase, where food enters the stomach and you get distension of the stomach. You get the intestinal phase, where various chemicals are being released and are traveling through the duodenum and the intestines. So importantly, you can trigger the acinar cells and the ductal cells directly through the vagus nerve. So in the cephalic phase, just by seeing food, smelling food, tasting food, this triggers the vagus nerve to come down, right? So let's say the vagus nerve's coming down and it says, yep, I'm gonna release acetylcholine to bind to the acinar cells, and I'm gonna release acetylcholine to bind to the ductal cells to trigger them to release their various components. So you can simply tell them to release not with the stimulation of peptides, triglycerides, amino acids, free fatty acids, and hydrogen ions, but simply just through the vagus nerve as well. That can also happen in the gastric phase. When the stomach fills with food, it distends. That can trigger the vagus nerve as well to do the same thing. But remember, that also triggers the stomach to release hydrochloric acid and chyme, which also triggers these as well. So the vagus nerve with these various stimuli are other ways that we can stimulate the pancreas to release its pancreatic juices from the acinar cells and from the ductal cells. So this is a quick overview of all the various exocrine functions of the pancreas. I'm Dr. Mike, and I hope that helps. Hi everyone, Dr. Mike here. If you enjoyed this video, please hit like and subscribe. We've got hundreds of others just like this. If you want to contact us, please do so on social media. We are on Instagram, Twitter, and TikTok at Dr. Mike Todorovic, at DrMikeTodorovic. Speak to you soon.